right, so good afternoon, everybody, and welcome to a program here with us at Exploring by the Seat of Your Pants. If you're joining us for the first time, and I know we do have a big new audience today, we are all about bringing conservation, adventure, and science into classrooms around the world through free live interactive broadcasts like this one. Now, today marks the capstone to a really special series we've had over the last week in conjunction with the amazing folks at the Gairdner Awards. So the Gairdner Awards are Canadian, and they celebrate and showcase some of the best biomedical and health researchers in the world. Many of their laureates, dozens of their laureates, have gone on to win the Nobel Prize, and we're always thrilled to have them as we showcase some of the coolest science at the cutting edge happening in the globe every single year. Now, over the last week, we've had the chance to learn about vaccine development, bioengineering, protein synthesis, and more, but we are wrapping up the week today with one, or starting off a new week. Sorry, it's our sixth program. There's so many programs I can hardly keep track. We are starting off a new week, one of our last weeks of the year, with Dr. Rodolphe Barrangou. So he is a CRISPR-Cas9 specialist. He is actually the Gairdner laureate who's joined us for the most broadcasts over the last couple of years. And today he's going to walk through some of his research and the field in general around CRISPR, this incredible genetic engineering tool that we have only really understood and developed in the last decade or less. I'm really excited to turn it over to him to showcase his work. I'm going to turn it over and stop talking soon so that he can highlight all the amazing work that's going on. And I look very forward to all your questions live and on YouTube at the end. So, Dr. Barrangou joining us in North Carolina, welcome to the broadcast and thanks for coming.
Jesse, thank you for having me. Good afternoon and good welcome. Good morning to most of you, as a matter of fact. Since you just mentioned adventure and science and wrapping up the week in style, I would argue there's no better way than talk about CRISPR and genome editing, and there are many applications and implications for society. And again, I likewise have to acknowledge and thank the Gairdner Foundation, the amazing people who work there, for bringing adventure and science to the classroom. So you've all heard about CRISPR, but everything you've heard about this week is on a continuum of what I call the innovator's dilemma. How do we bring science and adventure to the public? Right. And we have to translate our scientific understanding of biology, of bioengineering, of vaccines, of CRISPR, of genome editing, of machine learning, and on and on and on. And occasionally turn that scientific knowledge and understanding into technology that we can apply to eventually, hopefully, efficiently, and at scale, develop products that will be accepted by regulators, accepted by the public, accepted by the pundits, accepted by the media, and hopefully taught in the classroom, be that in middle school, high school, or even the university and beyond. So whether you're in California today or different provinces of Canada, I think we have Ontario and BC online here. I hope you have heard about CRISPR, and I hope that you will be using CRISPR before you know it in the classroom, and of course, as a consumer, and hopefully not as a patient, but if you need to, the technology will be there for you. Why am I here? Why did I get the Gairdner a few years ago with some of my colleagues, some of whom went on to win the Nobel Prize indeed? And why does it matter to everyone? And we're ushering in the second decade of CRISPR research, and we're observing in real time a revolution. And oftentimes you don't realize you're in the middle of a revolution until after the revolution has occurred. And as a matter of fact, you can see with the data here that this is a technological revolution unfolding before our eyes. 250,000 labs present in over 100 countries have collectively published over 20,000 papers written by nearly 50,000 authors that come from almost 5,000 different affiliations. So there's a lot of people working on this amazing technology to edit the genomes of organisms across the tree of life, primarily humans, human cells, and human models. And we can see that the whole world is working on CRISPR, including the US, first and foremost, China, number 2, and Canada, wrapping up our top 10. So what is it? How does it work? Why are we here? And why does it matter? Well, scientifically, CRISPR is immune system in bacteria, but the reason we're here is not the science of CRISPR. It's CRISPR, the amazing genome editing technology that allows us to alter DNA at will. And this is one of the reasons why the Gairdner Foundation recognized my colleagues and I a few years ago, as a matter of fact, 5 years ago, which is an eternity in CRISPR years. And the reason why people care about CRISPR is because it's an amazing genome editing technology that allows us to cut DNA precisely. Think Control F, right, in a Word document that would contain the 3 billion letters that are in our genome, we can very precisely, very selectively, very efficiently look for a string of letters and then selectively slice DNA, cut DNA precisely at that location. And then following cleavage, the DNA will repair itself, patch itself up essentially, and then generate a mutation precisely at the site of cleavage. For instance, here turning a G into an A. This is what is called genome editing. We can edit the DNA by selectively geolocating a CRISPR effector at a precise location in a very long string of A, T, Cs, and Gs, and then trigger an edit. Now, as a matter of fact, in the last 10 years, my colleagues and I, and again, 50,000 different authors across over 100 different countries, have been very hard at work sharpening the CRISPR toolbox, enabling us not just to change the DNA sequence, but also the DNA structure, not just changing the letters, but changing the punctuation and managing how much or whether genes are turned on or turned off, turned up or turned down, functional or dysfunctional, written as they were when we first came out, or corrected and rewritten as an editor would a typo in one of your essays or homeworks. So 10 years later, we now have a toolbox that enables us to virtually, efficiently, specifically, and at scale, modify the DNA, the RNA, and products thereof across any organism on the tree of life. Well, what does that mean? It means that this is what I call the CRISPR zoo. In the past 10 years, those 50,000 authors have been very hard at work showcasing how we can use CRISPR to manipulate the DNA of the smallest entities on the planet, like viruses, think COVID or HIV or bacteriophages, bacteria, yeast, and model organisms like historically C. elegans and Drosophila, all the way to non-humanoid chimps and some plant models. And all academics working on those organisms have used that technology to understand and decipher DNA and its role in function in entities and phenotypes of interest to us. Now, in the real world, including in food and ag, people have been very hard at work using CRISPR to rewrite, correct, edit, alter, manipulate the DNA of crops. Think corn, think soy, think rice, vegetables, think tomatoes or mushrooms, seafood, obviously CRISPR shrimp, or even livestock, think CRISPR chicken or CRISPR beef or dairy for milk products, so to speak. And then that same concept can be used to edit the DNA of organisms and or cells and or viruses of interest to medical applications. Whether it's a human, whether it's an organ, whether it's a cell, or whether it's a tissue, we have the ability to use CRISPR to rewrite that DNA. So let me show you one example of what can be done with CRISPR. Adventure and science in the classroom. People can take cells from a butterfly like this, target a gene called yellow, which predictably is responsible for encoding the color yellow in the wing butterflies, in the wing pattern of butterflies, and we can grow a smaller butterfly out of those cells that are edited with CRISPR that will grow with the same exact wing pattern to the smallest details you could see and decipher. And the wing pattern of a butterfly is very much like the human iris. It's unique across the world. No two individuals have the same molecular pattern, except that we took out the color yellow. And you can do that with other colors. The point here I'm trying to show visually is that we have the ability to use CRISPR to recolor the pattern of winged butterflies. And if we can do that, I would argue there is no limitation technically, but our imagination to what we can do with CRISPR when it comes to editing organisms across the tree of life. Now, CRISPR is now a 10-year-old, and it's the most promising, amazing, disruptive, revolutionary 10-year-old we've seen in biology in a long time. And the reasons are many, as you can see here: programmable, specific, transferable, efficient, precisable, affordable, quick, multiplexable, and scalable. But it's only 10 years old. So if you look at technology that you use at home or at school, right? The phone 10 years in did not look like this. The plane 10 years in is not something, a vehicle or contraption you want to get in, or first submarine, or the first NASA shuttle, right? 10 years is a very short amount of time for technology, think the internet or computers and the like. Nonetheless, this 10-year-old has been amazingly scaled up, but it's still imperfect. So people are still working on making this tool as sharp as possible, as efficient as possible, as specific as possible, as deliverable as possible. And what CRISPR has made is enable people to virtually change human DNA however we wish for medical purposes. But there's a lot more than we can do with CRISPR. So think about all the products and applications we could do. So people can use that technology for organisms of interest to biotechnology. I know you heard about bioengineering earlier this week. So think about bacteria, yeast, algae, fungi, all kinds of organisms, simple or complex, that are involved in the food manufacturing process, the bioproduct manufacturing process, household care. Think of detergents and soaps, or bioenergy and biofuel. All the organisms used to cross the tree of life to make those biotech products, those biological products made by cells and living organisms, have had already their genome enhanced, sharpened, improved. Their efficiency, their energetic output and throughput improve with CRISPR. The same is already occurring in the ag world. So think of crops, think of livestock, think again of microbes, bacteria, yeast, algae, fungi, and the like. Think of trees, think of flowers, think of grasses, think of seafood, think of aquaculture, crustaceans, mollusks, and the like. Many of those organisms have had their genome enhanced, altered. They have had molecular breeding with CRISPR, but most of them are not quite as of yet on the market, but some are. I'll discuss that in a minute. And then, of course, at the very top of the value chain, maybe of the food supply chain, humans, with therapeutics, gene therapies, cell therapies, antivirals, antimicrobial, altering the microbiome, altering the virome, cell therapies, immunotherapies, engineering humanized tissues in animals to have xenotransplantations from one species to the other, gene drives, diagnostics, pet care, vet care, on and on and on and on. The possibilities are limitless. Now, let me give you a very specific example of how far and how fast CRISPR has made it to the clinic. So if you've never heard of Victoria Gray, you should Google her, right? She was the first patient in the US who was able to receive a CRISPR trial experimental therapy for sickle cell disease. And earlier this summer, she had the 2-year anniversary of the single dose that she received that essentially cured her sickle cell disease, SCD. And what they did is they took some of her own cells out of her own bone marrow, took them into the lab, used CRISPR to target a broken gene responsible for sickle cell anemia. They corrected that typo in her genome, expanded those cells in the lab, confirmed everything was fine and safe and as expected or hoped, and then reinfused those cells into her body as novel genome edited corrected medicines. That approach is being used in dozens of clinical trials, including some that are being managed by the FDA in the US, but some have already gone underway in Europe and elsewhere to address human genetic diseases and correct human genetic diseases. And there is about 800 different known human genetic diseases in the literature thus far. And last year, Intellia, one of the companies I co-founded actually, showed that we can use CRISPR in vivo in humans, just deliver CRISPR to the liver of a patient with a liver disease to correct their DNA and tone down a disease that was responsible for degeneration. That same technology, according to the World Economic Forum, is poised to revolutionize farming. It's not me saying it. This is the most authoritative economic engine on the planet, saying that next generation biotech and genomics in general, and genome editing in particular, are anticipated to generate up to $100 billion in additional farmer income in the next 8 years, to produce an extra 400 million metric tons of food and reduce malnutrition for up to 5% of the population. What they anticipate, as a matter of fact, is that up to 15% of all the farms in the world will have planted a genome-edited seed by 2030, and the number equates to 100 million farms. Think about the scale of the endeavors that we're discussing here. This is how real this is. We're talking about all the crops you can think of: wheat, soy, rice. We're talking about crops for vegetables, lettuce, kale, mushrooms, tomatoes, potatoes. And of course, we're also talking about trees. We're also talking about outside of the plant world. We talk about beef, dairy beef, or animals that we use for milk. Not just cows, right? We think about sheep and the like, llamas, whatever you can think of. Of course, swine and pig for pork. And of course, poultry for eggs and or for their meat. And of course, I mentioned bacteria, yeast, algae, and fungi that are there. The technology has been advanced enough in the real world already that hornless cows have been bred and born, and second generation are there. So we have a more humane, more sustainable food supply chain. Same for cheese. The bacteria used to make yogurt and cheese across the world have been naturally CRISPR enhanced for phage resistance over the past 11 years. CRISPR tomatoes are already on the market. CRISPR edited tomatoes are on the market in the UK and in Japan. And the New York Times covered maybe the future of food, where we can use CRISPR and genome editing to breed a healthier, more resilient, more sustainable food supply chain. And of course, it's 2022, we're on the cusp of 2023, and there is no bigger challenge, maybe no bigger endeavor or more noble pursuit than using CRISPR and genome editing to breed a healthier forest. You can think of all the wood used for fiber and timber in Canada and the US and beyond. Biofuel, bioproducts, densify wood alternative to plastics, hydrogels, glues, foams, food and feed, and of course, bioremediation. Of course, carbon capture, carbon storage, and of course, managing the land as best as possible at a time where drought, heat, and extreme weather are a challenge for a more sustainable, more efficient, more resilient agriculture in general, and forestry in particular. And of course, I presume your imagination is titillated here. Hopefully your thoughts are provoked to some extent. Again, adventure in the classroom, but think of the scales in play here. We can target something as small as a virus, invisible virus, a microscopic bacterium, an edible plant, a sick human, or a healthy tree. What we use to do it, what kind of edit we want is a little bit more technical than I want to go into today. But think about the size implication. We can change things like a single letter. 10 to the 0 is 1, right? Your math teacher would be pleased with that. And we can change sometimes to make deletions that are as large as 100,000 letters in a genome. We can change genomes that are very small viral genomes, or genomes that are very big. The human genome is 3 billion letters. Some of the tree genomes that we work with are 30 billion letters. In some cases, it will be one small dose to one patient to kill one type of cell. In other cases, it will be a very large edit for a very large tree that will live for hundreds of years and not just capture carbon to benefit people who are here now, but people who are not born yet. Think of some of those trees benefiting your kids or maybe even your grandkids and great-grandkids. We're working on this right now, right here. But with great hopes, great aspirations, great expectations, also come great responsibility. And, doing science is great. I'm a passionate, active scientist. I'm a professor, an entrepreneur, the editor-in-chief of the CRISPR Journal. There's a lot of things I do all day, every day. But we have to understand not just the applications, but the implications of our work. How is our work perceived? What are the ethical implications of editing people? Are we going too fast? Are we going too slow? Are we being responsible? Are we being cautious? Do we have a sense of urgency, responsible enough to address climate change now and patients that are dying, but also have the wisdom to be patient enough to make sure that the way we do it is the most responsible way to do it right here and right now. Of course, there's a lot of anti-science bias, misinformation, and disinformation. You heard about vaccines earlier this week. I'm sure there's a lot of relatable issues here. And we have to do that also for a technology for which first-generation tools to do genetically modified organisms was poorly marketed, poorly communicated, poorly managed, poorly regulated. And there's a lot of skepticism there as well. So we have to build narratives. We have to have voices of patients and farmers and foresters and mothers that can explain to the world why we're doing this, not how we're doing it, and who is doing it, not where we're doing it. This is why saving lives, having a more sustainable agricultural system, having a safe, efficacious, health-promoting, plentiful food, and to do so in a very complex geopolitical system where food safety, drug accessibility, diversity, equity, inclusion are such challenges that we have to solve the political problem, the economical problem, and the scientific problems. If you're curious about this, I would urge you to read the CRISPR Journal and some of the great papers published by those 50,000 authors in 25,000 papers about how to manage this. How do we democratize that technology? How do we share that tool with people who hardly can afford it? What are the milestones that we need to bring that to the clinic? What are the ethical implications? What are the stories that we should tell? How do we get buy-in from the regulators and the courts and the system and the lawyers? And this is why no matter what pursuit you want in your own life, whether you want to be a scientist or not, a technologist or not, an engineer or not, whether you want to go to get a PhD or not, an MD or not, you will have a role to play in this. Of course, academics will do the science, but we have to work with government, we have to work with industry, we have to work with society. We need media influencers to explain to people what CRISPR is and how it works and why they should care and why it matters and why it will benefit their lives. We need to work collectively. We need to work together to do so. And the challenge is that we have to get and learn to wear different kinds of hats to play our part, whether you're a researcher, a teacher, a speaker, an entrepreneur, a learner, a coach, a writer, a thought leader, an administrator, an ambassador, a spokesperson. You all have a role to play in the promotion and advancement of science and technology to solve the biggest world's problems. And this is why I'm so thankful to the Gairdner Foundation, right, for the opportunity to talk to you today, because I know that some of you will relate to this. I know that some of you will want to invent next generation technology through research. I know some of you will teach the next generation of scientists. Think the people in your classroom today teaching you how to do this. We'll need great storytellers, right? Influencers. We'll need great innovators and entrepreneurs. We need great recruits, talent, and people who will do the work. We need people to educate investors, to educate inventors, to educate the governments and the regulators and the public and the media. We need to tell those stories, and we need all people like this to share the gospel of science. People like Tony Fauci, people like Jennifer Doudna, people like Feng Zhang, people like Emmanuel Charpentier and my colleague Philippe Orvathier or Dr. Plummer, the late Dr. Plummer. And this is why we invite you all to also educate yourself about the Gairdner Foundation, right? Not so long ago, one of our colleagues and laureates earned the Nobel Prize. Tony Fauci, Saint Fauci in many ways, has played a critical role in communicating about the trials and tribulations of the scientific community at times where we need a moment of science. And then, of course, the late, great Frank Plummer, who was very important in managing infectious disease in Africa over an illustrious career. We all have to play our part and do what we can do. So I'm going to thank you all at this point in time. I'm going to stop sharing my screen, and time permitting, we'll have a few minutes for Q&A from the various classes, and Jesse will be our MC.
I will try my best. That was a spectacular program. Thank you so much, Rodolphe. As usual, I wanted to put in the chat for everyone the YouTube link to Human Nature. If you want to see the trailer for it, it's spectacular. I'll put it in our YouTube chat for everyone as well. But a really, really spectacular documentary if our classes get the chance to check it out. I don't think we're going to be in short supply of questions, so I'm not even going to share any of my own. I'm going to go straight to Ms. Ball's class joining us in Goderich. If you have a question to kick us off, you are good to go. Welcome in. Hey.
Hey, Jesse. I think we were just wondering what the biggest myth or misconception you feel is about CRISPR.
Yeah, I think the biggest myth or misconception is that it's dangerous, right? I mean, it is a solution, not a problem. And most people, the large majority of the scientists in the world, using this technology to bring it to solve problems, are trying to cure human genetic disease. And there's a lot of hype and mishype and communication about human enhancement and more intelligent humans and on and on and on. And right now, it's hard enough just to cure disease where we know a single gene with a single base, and if and when corrected, will cure disease. That's still hard to do. It's not trivial whatsoever. Delivery, efficiency, cost, manufacturing is important. That's what we're spending our time on. And if you watch the movie, which you all should, Human Nature is amazing, right? People are worried about human enhancement. Are we going to use CRISPR to breed more intelligent humans? And we don't even know what intelligence means. We don't even know what genetic and environmental factors are responsible for the enhancement of human intelligence. So I think there's a lot of concerns about the misuse and abuses of CRISPR. You can look at the CRISPR baby drama stories from Junqui Hay, who was released from jail, as a matter of fact, earlier this year after serving a 3-year prison sentence in China. I'm not going to comment on that, but it's intriguing. So there are risks for misuse and abuses, but the biggest urban legend is that scientists are spending their time trying to enhance humankind with just trying to solve disease and feed the world.
Great question, Ms. Ball. I want to note, too, just as books for people to follow up with, Crack and Creation, Jennifer Doudna herself, and The Codebreaker, Walter Isaacson, detailing the whole field in general, is just our spectacular reads for classes that are interested in keeping the learning going. We're going to head to Notre Dame High School, Ms. Gerard's class. Welcome in Belmont. If you guys have a question for us, come on in.
Excellent. Thank you. Come on. Okay, Sarah.
Could you elaborate on any ethical controversies involved and aligned with CRISPR and any dangers that can arise from using it?
Yeah. So this is a great question, very relevant, very timely. As a matter of fact, we had a whole issue here thinking about CRISPR, right, of the ethical implications of this. So let me just mention two. The first one is germline versus somatic cells. So if I get some of my cells edited, somatic cells are the ones that I'm not going to pass on to my progeny. Germline, in my case, sperm, is what I'm going to pass on to my progeny. So there's a lot of controversies about whether we should or not allow germline cells to be edited. There are some extreme cases where it's ethically defensible and applicable, but the challenge is that we should not have the power to make decisions for people who are not born yet, and to make decisions for the next generation. So that's a sensitive topic. That's a controversial topic. And then to some extent, this also applies to patient consent, right? Do patients really understand what they're consenting to? Do they really understand and appreciate the risks and implications for them and for their loved ones, or for them and their offsprings? And ensuring that we are ethically transparent and responsible enough to ensure that the patients understand what the ramifications are for them is still a challenge, is very important, and is not universally equitably implemented throughout the world because of different regimes that manage medicine in different ways throughout the globe.
I'm bringing this up in the chat right now, too. Now, this is actually written before CRISPR was a really big deal in the world of sort of biology. There's a really fantastic book talking about, in general, genetic engineering and some of the ethics around it, and how the field has really policed itself in a really big way, in a very noteworthy way. The Gene by Siddhartha Mukherjee is just a spectacular book detailing this, the whole history of genetic engineering for the last 50 years now, since the 70s and beyond. So I encourage classes to check that out too, to sort of detail some of those things. I've also put a link in the chat to the CRISPR journal. So if you want to read that ethics article, if you want to learn more about what's going on there, see some of these covers behind Rudolph there, do check that out if you get the chance when you're done. Ms. Zimmer's class, I'm heading to Oakville to come to you guys, and then I'll do another round of questions with all our live groups. But Ms. Zimmer, you should be in. You're good to go. Audio.
Great, thank you. We've got our student coming up with her question. One moment, please.
Hi, can you hear me?
Yes,
Yeah.
they can hear you.
Okay, so I thought you mentioned about like genetically mutated foods and how it saves lives. So I was just wondering, are there any like consequences in eating genetically mutated foods, and maybe the effects of eating it and how it affects the consumers like in the long term?
Absolutely. So there's a great report that was written by the National Academies of Sciences that came out a few years ago on the safety of biotechnologies in general and the impact of genome editing and manipulation and GM technologies in the food supply chain in particular. It's a 400-page report that I very strongly recommend. I think it's a great read. It can be tedious, laborious at times, but the bottom line is that for decades, we've been able to document the absence of negative impact on both the environment and humans as a species of consumers when it comes to the consumption of genome-edited foods. And if you look at what we're doing now with CRISPR, our ability to do that more efficiently, more specifically, more accurately, and at a smaller scale that's more surgical, is opening up new avenues to do so, not just more responsibly or more efficiently, but more multiplexibly. And that means that for the food supply chain in particular, what we're working on is not just traits of interest to farmers. Think of increasing yield or pest resistance or pest resilience or drought resistance and the like. But we're also now, because we're able to do it more efficiently, going to benefit the consumer in a more tangible manner. So the point is that we're going to make food more nutritious, which means that when we consume it, we're going to have enhanced health, right? And I think that this is where there's a lot of skepticism about, well, what's the impact on the environment? What's the impact on the consumer? If I eat something that's genetically modified, am I going to be genetically modified? There's a lot of scientific nonsense out there that I'm sure you get debunked in the classroom. But the evidence, the data, the information by the most scientifically sophisticated society in the world has written a report and concluded unequivocally based on the data, in the absence of any conflict of interest, that those technologies are safe to consume, safe to disseminate, and are not of a negative impact on our health. And the point about CRISPR now is that moving forward, we're only going to not just do that and make it more sustainable, more efficient, more resilient, but we're also going to enhance the flavor and the texture and the chemical composition of those foods so they are more beneficial to our health. So think more nutrition. I mentioned nutrition. Think more nutritious food. Think that the ions that we need, the molecules that we need. Think of the benefits of a good French wine, for example, being packaged, repackaged, or enhanced in grapes, right? So we have even more benefits of that without having the caveats of an alcoholic consumption. So there's a lot of applications. Again, I've written a piece on this for the National Academy of Engineering issue from last summer 2022. I'll post a link when I find it, or have Jesse find that as I'm speaking here. And then you can educate yourself on that topic as well. But great question, very important story to tell.
I was finding all sorts of other things, so I'll have to find that one in a minute. But I did put a link in the private chat to the National Academy of Sciences paper, so if people want to check that out. One of the things that's really interesting about sort of genetic engineering in foods is that some countries are really willing to try and see what would happen, basically, with understanding that there would be no reason to suggest they'd have negative impacts on health, and some countries outright ban them. And so we've had this like many decades of sort of seeing how it played out. It's like with COVID, we got to see not only the tests and trials to understand how the vaccines would affect people, but billions of people use them in a very short period of time to be able to say, look, the data is really clear that this is safe. And so we've seen a European-American difference over this in the last few decades. So it's been really interesting that this is a topic that keeps getting brought up over the last few decades, but that there's no indication that it has any negative impacts on people. A great book, or great two stories to keep the learning going with this. The Wizard and the Prophet by Charles Mann, one of my favorite science writers in the world, details some of the work to improve crops, improve crop yields, make them tastier, better, healthier around the globe. So I encourage people to check that out. And specifically, Golden Rice as sort of an analogous, a story that students should look up about the benefits of some genetic engineering when it comes to a food, and also some of the pushback on it because it was genetically modified and how that impacted human health in a lot of countries around the world. So Golden Rice is a really great story for classes to check out when they're done. Let's do another live round of questions. Ms. Ball's class, I'm coming to you first. Come on in and take us away.
I just wondered if you could highlight a few diseases that we've either used CRISPR already for or that we're excited about using CRISPR for.
Yes, sickle cell, anemia, beta thalassemia, and TTR, transthyretin amyloidosis. So in the case of sickle cell, it's oxygenation in the blood, right? There's a significant portion of the African-American population that suffers from this genetic disease, which conveniently in nature gives them resistant to mosquitoes that spread malaria. So there's a rationale for it, but in today's world comes at the cost of poor oxygenation in the blood and shortened lifespans, because the shape of the red blood cell isn't the shape of a sickle cell, which has all kinds of circulatory issues. And this is the story of David Sanchez in the Human Nature movie that is up there in the link. So that's a very well-documented genetic disease. That's a very well-documented mutation that we know how to correct and edit using CRISPR. And this is what Victoria Gray is all about, what David Sanchez is all about. This is well chronicled and documented, and it's getting far along in the clinic, whereby multiple CRISPR-focused companies have started multiple clinical trials right now throughout the world in 5 different countries, I believe: Australia, the US, the UK, which is not part of Europe anymore, the EU, and I think Japan. So there's a few there. Beta thalassemia, hypercholesterolemia, Leber amaurosis for the eye. Those are like the second generation that are getting in the clinic right now, with some patients have been dosed, some therapies have been advanced. A lot of preclinical work has been done in mice and animals and human cells, and now we're getting to dosing patients. And then moving forward in the future, really cancer is what a lot of cell therapies are all about. There's news that came out just this weekend from a single patient CAR T therapy, so CAR presenting antigen engineered cells to trick the human immune system into targeting cancer cells in particular and curing cancer. In this particular case, leukemia, but this is opening the door for all kinds of cancers that are out there, all kinds of tissues, all kinds of applications and the like. So, and we also, last but not least, are working on antivirals to target dormant or silent viruses in our tissues. You can think in this case, HIV, and obviously COVID is the primary focus of interest for the antiviral applications of CRISPR.
Yeah. I love one of the ethical discussions that's come out around these sort of therapies in the future, that we can get rid of some of these genetic diseases. And that is, if we have the technology to rid the world of something like beta thalassemia or sickle cell anemia or cystic fibrosis, why would we, is it unethical to not do it if we have the technology to do it? So these are some of the discussions we're going to be having, and please elaborate if you want to talk more.
Yeah, I mean, my colleague George Church says not using CRISPR to address genetic disease is like having a fire extinguisher in the middle of a fire, not using it, right? So I may not go that far when it comes to irresponsibility, but some people do feel that way, and the power of that technology, the use of that technology, the short-term potential of that technology is so big that it's almost, I would argue, irresponsible not to use it to address the biggest medical and environmental challenges that we have lying before us. And we know there's too many of them right now. So we need a moment of science and use the best technology possible to solve the biggest challenges possible.
Very, very cool. We have time for 2 more questions. Time flies when you're having fun. I know we're at the 40-minute mark, so I will put up some just resources for our classes. Again, do check out Gairdner. I'll put this up at the end of the broadcast as well. Kraken Creation, Codebreaker, at CRISPR Lab, Rudolph Showcase, and Human Nature, of course, is a documentary. But let's head back to Notre Dame High School and then Madam Zimmer to wrap up the broadcast. Come on in and Belmont, guys. Hey.
Hi. With altering cells to target viruses or sick or infected cells, how does CRISPR technology or CRISPR decrease the possibility of autoimmune disease when altering cells to target viruses or sick and infected cells?
Yeah, so autoimmune disease have their own challenging issues that would take more than 30 seconds to give a take on. But the way the antiviral applications work, again, quickly, is there's different life cycles of a virus that can manifest themselves into a human cell. There's some what's called free viruses, right? So where you have like a virus floating around the cell, we're going to bring CRISPR in and then cut that viruses and kill it so it can't replicate anymore. There's a more pervasive challenge of viruses that are integrated into our genome, integrated into our chromosome, that lay dormant for a longer time, inserted proviruses. Those are much more tricky because they get passed on, they get silent, they're invisible, they're not as pathogenic, pathological, but they can be induced in different conditions, environment, and cues. And what we do in this case is we have to encode CRISPR to come in on both sides and excise that virus very efficiently. The tragedy is that we are very good at doing that, and we're almost 100% success rate. We can take out a virus outside of a human cell. The challenge is that we don't have the delivery modalities yet to do that in 100% of the cells in the whole patient. So until we find ways to deliver this technology more efficiently, specifically and at scale, I can't take a patient and dunk them into a pool of CRISPR and come back a week later and see if it worked. We have to be efficient. We have to be quick. We have to be able to have the right dose. And we're working on that. And then we'll get to autoimmune diseases later on and repeated doses and responses. And so we'll get there. It's a good question, but it's not a first generation question.
Is there any, are there any CRISPR journal articles or stories that detail this in any bigger way if students were keen to follow up? Or
Yeah, so we have a whole issue of CRISPR's path to the clinic that we published earlier this year.
There you go.
This is going to be big in 2023. So just follow the news and you'll be up to date for sure.
It's a nice thing for students in today's classes. Like, I mean, they've grown up where this, sort of came into being, and it's changing month by month. I mean, as you detailed at the beginning of the presentation, there's new research every single day. So if you are excited about this topic, if you want to make your mark on this field in general, now is the perfect time to be in grade 11, in grade 12, interested in this going into university, because there's so much development and excitement in the discipline. Madam Zimmer, we're going to come to you for one last question. You're in the broadcast. You're good to go, and we'll wrap up after that. Thanks, guys.
Wonderful. Thank you so much. During your presentation, you had listed or you had posted 2 books. I'm wondering if you could put that in the chat for our class, please. And we have our next student who's ready to ask her question. Go ahead.
Good afternoon. My question is, who or what organization finances the CRISPR gene editing project?
So pretty much everybody is investing into CRISPR, right? The public can invest by buying stock and going into those companies, and investors and venture capitalists fund a lot of the work that's going on in the industry. Governments invest a lot, NIH funding, NSF funding, Health Canada funding, depending on where you are. The Chinese Academy of Sciences is putting a lot of money into CRISPR as well. So governments invest. And then not-for-profits. Think of the Bill and Melinda Gates, the Novo Nordisk Foundation, right? The Moore Foundation and others, not-for-profit organizations who want to ensure that there's an equity piece to it, accessibility piece to it, and the like. So the point is, the public, the investors, the bankers, the market, the street, right? Wall Street, the companies that are investing in the technology themselves to work with other companies to do that, big pharma, big ag, big biotech, governments, and not-for-profits. Everybody wants a piece. And it takes that kind of effort and commitment. We're talking about billions of dollars already invested into CRISPR in the past decade to get to where we are right now. We're going to need another $10 to $15 billion to bring that all the way into, half a dozen therapies and the top 5 crops and the top 5 most industrially relevant forest species.
I'm trying to think of another field in which there's been so much excitement so quickly and so much investment from so many different sources. The only thing that really comes to mind is the vaccine development and the speed during the pandemic. But that's about it in my life.
AI is happening right now, changing the world, right? With the internet at the time. I think there's a few examples, but they're few and far between.
Yeah.
And CRISPR is a great example of how much and how fast and how far the world is changing.
Well, thank you so much for being our guide to a lot of it today over the last 45 minutes. Again, we've shared a bunch of resources in the chat. Any one of our classes can follow up with us if they want more detail on any of these things. Rodolphe, it's always a pleasure having you. I really encourage our classes to check out the Gairdner website, see some of the amazing laureates they've had over the last 2 years. And if you want to see any of our programs in this series over the last week, you can head to our YouTube channel. Oh, that's not our YouTube channel. There's our YouTube channel to check out any of the broadcasts. You can check out this one. You can share it with your friends and family. Anyone who's tuning in, very easy to keep the learning going. And as you know, what we do to wrap up every broadcast, I'm going to bring in our classes to say thank you and farewell. Miss Bowles' class, Notre Dame High School, Madam Zimmer, thank you guys for joining us today. And have a wonderful rest of your day.